What is the role of insulin? The master regulator of blood glucose levels, able to stimulate glucose uptake by adipose tissue and muscle, all while suppressing the release of glucose from the liver. Right? Correct. However, as well as regulating glucose homeostasis, insulin also induces fat storage and fatty acid uptake in adipocytes, thus ensuring sufficient storage of energy that can be mobilized during fasting when insulin levels are low. Obesity disrupts the balance of energy storage and mobilization, and over time, tissues normally responsive to insulin show reduced sensitivity, termed insulin resistance. The elevated level of circulating free fatty acids in obese individuals is thought to be a significant driving factor in the development of insulin resistance. This is due to the fact that fatty acids induce endoplasmic reticulum stress, cytokine production, and activate the c-Jun-NH2 terminal kinase, or JNK, signaling pathway, all of which play a role in mediating reduced sensitivity to insulin. Additionally, in response to caloric overload, adipocytes expand, and a pro-inflammatory response is mediated. This involves the secretion of tumour necrosis factor alpha and monocyte chemoattractant protein 1, which drive macrophage accumulation into adipose tissue, again contributing to insulin resistance. Upon binding to its receptor, insulin triggers intracellular signaling cascades that have both metabolic and mitogenic effects on target cells. The pathway responsible for control of cell metabolism is the PI3 kinase pathway. Once activated by insulin, it promotes glucose uptake in target tissues, as well as the suppression of cell death, whilst the MAP kinase-mediated insulin pathway promotes growth, differentiation, and survival. In a state of insulin resistance, activation of the insulin PI3 kinase signaling pathway is reduced. Thus, glucose uptake via GLUT4 transporters in target tissues is reduced, and blood glucose levels rise. The pancreas responds to the rise in circulating glucose by increasing insulin secretion. Compensatory hyperinsulinemia restores function of the PI3 kinase pathway, thus glucose uptake and gluconeogenesis in target cells are normalized. Researchers have found that cancer cells are able to convert this glucose to protein, DNA, and even lipid triglycerides, as they are able to harness a metabolism similar to that of adipose cells. Although the high plasma concentration of insulin successfully normalizes activation of the PI3 kinase pathway, it leads to hyperactivation of the MAP kinase pathway. This selective imbalance of the two signal transduction pathways leads to excessive proliferation and growth, thus has tumour-promoting effects. In addition, due to the considerable homology between the insulin receptor and the insulin-like growth factor 1, or IGF-1 receptor, insulin can also bind to and activate the IGF-1 receptor. Signaling via the IGF-1 receptor is also linked to cell proliferation and survival. Thereby, hyperinsulinemia also upregulates this tumorigenic pathway. Hyperinsulinemia also leads to increased secretion of IGF-1, in part by suppressing the production of insulin-like growth factor 1 binding proteins, IGF-BP1 and IGF-BP2. This excess IGF-1 can hyperactivate IGF-1 receptors and insulin receptors, again stimulating proliferative and anti-apoptotic events in both pre-malignant and malignant tissues. Hyperactivation of IGF-1 receptors are also thought to reduce the expression of E-cadherin in the cell membranes, thereby promoting cell migration. Epidemiological studies have shown that elevated circulating insulin and IGF-1 levels as a consequence of obesity are associated with several cancer types. These include colorectal, endometrial, pancreatic, liver, premenopausal and postmenopausal breast. For example, In 2003, Lucanova et al. Performed collaborative case control studies in New York and Milan. They reported that females in the highest quantile of serum C-peptide, a surrogate biomarker of insulin, with a BMI above 30, had a 2.57 times increased risk of developing endometrial cancer.